This is the unusual inheritance mechanism genetics tutorial presented by the SchrDbb reference ABC of Clinical Genetics by Helen Kingston. So here we're going to visualize the other inheritance mechanisms. So the other inheritance mechanism, you can visualize it on this table here. So we need to know that what we have already spoken about the chromosomal inheritance method, and we have spoken about the Mendelian inheritance method. Now we are going to speak about other inheritance method, other mechanism of inheritance. So other mechanism of inheritance are mostly due to point mutations. Is it clear? So we have these are other inheritance methods due to point mutations. So point mutation is because of a mutation in the nucleotide. So we have an addition, a removal of a nucleotide, or we have addition and removal of a codon is going to produce those point mutations. So that is what is called point mutation. So on point mutations now, we need to see, so these are other unusual inheritance methods. So one is point mutation. So as you can see here, this is so we have gain of function of mutation is due to the CA gene repeat, repeat gene. So CAG is a codon, C is cytosine, A is adenine, and G is guanine. So when you have a repeat, normally if this CAG codon repeat for 6 to 35 times, it means that you are not going to express Huntington's disease. It means that you are normal. So that CAG repeat is normal when it is expressed 6 to 35 times. And now if it's expressed now 36 to 100 times, so if you have a repeat of 36 to 100 times, you are going to express Huntington's disease, which is which is going to be expressed in another form as a point mutation or a trinucleotide repeat expression. Is it clear? Because I've said that in point mutation you may have deletions or you may have additions. Is it clear? You may have deletion or you may have addition of nucleotides, and this is what happens. Now in Kennedy syndrome, normally you have a repeat of 9 to 35, and if it increases to 38 to 36, you are going to have Kennedy syndrome. In spinocerebellar ataxia, normally you have a spinocerebellar ataxia 1, 2, 3, 6, and 7. These are the different spinocerebellar ataxia. And when you have a repeat of the CAG gene, CAG codon for this number of times, you're going to have that. And this is the last disease, the dental pallido-louisal atrophy. So now you have now repeat expansion, other repeat expansion, like in the cases of the CGG gene. So you can have a fragile X syndrome. So when you have a repeat of this number of times, you have a fragile X syndrome at the E site. These are the site A. You have this still. So these are the different order. Friedreich ataxia syndrome. You have myotonic dystrophy, and that when you have an addition of this number of the CTG gene and the CTG gene respectively for this number of times. So you see, this is the other method of inheritance can first be a point mutation where you have a repeat gene, an addition of the particular codon on the gene set. Now the second method of inheritance can also be due to a uniparental disomy. You can have a point mutation. The second method, it can be due to a uniparental disomy. Now, what is a uniparental disomy? I've said that uniparental disomy is a transmission of the chromosomal disorders, transmission of genetic diseases via the grandparents' genes. Is it clear? Transmission of chromosomal disease via the grandparent genes. So this is another unusual mechanism causing human disease is uniparental disomy, where both copies of particular chromosome are inherited from one parent and known from the other. So are you getting? So here you have inherited gene only from one parent or even one grandparent. Is it clear? So and the other parents do not inherit. Now this autosomal uniparental disomy usually, so when you have a trisomy, so when you have, in this case you are not going to have a trisomy disorder, because the uniparental disomy is going to be produced by what is called a trisomy rescue. Is it clear? And if there's a trisomy rescue on the good chromosome, if you have a trisomic rescue on the good chromosome, then the patient is going to develop a uniparental disomy. And there are two types of uniparental disomy, as we're going to see later. So this is a table just showing uniparental disomy. One parent is the father and another parent the mother. So you see that the father is going to have all these chromosomes, is always going to divide to produce these gametes, the mother is going to produce this gamete. Is it clear? So you see that when this father has these gametes, yes, and this mother has these gametes, so this is going to fuse. This one has done non-disjunction. Is it clear? So it's going to fuse now to produce that. Now if there is a trisomy rescue, so here you are going to have a trisomy. Are you see? So the father chromosome is going to fuse with the mother chromosome, which has done non-disjunction. You see that there are two chromosomes here, while in the father there is only one chromosome here. Is it clear? And you see that the genes here is with the gene of one grandparent. This mother here had the gene of the fathers of her father's grandparent and of her mother grandparent of her mother. Is it clear? So you see that the gene that this child is going to have is a gene of the grandparent. Is it clear? You see the difference. That's why I was trying to say that you in uniparental disomy you receive the gene from the grandparents, and that gene from the grandparents either is coming from only one parent. Is it clear? So this is the gene of the grandparent. When you have the trisomy rescue on the gene of the father only, you are going to have now the gene of only the grandparents in you. Is it clear? And from only one parent. So this is uniparental disomy, and this form of uniparental disomy is called heterodisomy. Is it clear? Heterodisomy is because it is from both grandparents. It is a homozygous pair that has been produced, heterodisomy. Now this heterodisomy is produced when you have non-disjunction at the level of meiosis 1. So when you have non-disjunction at the level of meiosis 1, you have heterodisomy. So let me explain again. This is the father, this is the mother. So there is non-disjunction meiosis 1 in the mother. Is going to produce two chromosomes which have done, and one chromosome is going to produce two cells with the chromosomes not separated, while the father have separated the homozygous pair. Now there's a fusion to produce a zygote. If there's trisomy rescue now on the normal chromosome of the father, you are going to leave only the zygote now with the chromosomes of one parent, who is the mother, and that chromosome of one parent, who is the mother, is constituting of the chromosome of her grandparent, of the grandparents of this child, or the parents of this mother. Is it clear? Now when you have the chromosomes of both the grandfather and the grandmother in a homozygous pair, we call it heterodisomy. Now if you have a chromosome, as you can see here, of only one grandparent, let's say the grandfather, it is called isodisomy. So let's see again this pattern. This is the father, this is the mother. The father has done complete separation of the gametes, with all the chromosomes being separated. The mother has done again now non-disjunction at the level of meiosis 2. To produce a heterodisomy, it was non-disjunction at meiosis 1. But to produce an isodisomy, now the mother has done a non-disjunction at meiosis 2 to produce gametes now, and this gamete now is only going to constitute of one grandparent chromosome, let's say the grandmother's chromosome. So now when there is fusion of the father and the mother's gamete, they are going to produce a zygote. And let's say that there is a trisomy rescue on the normal father's chromosome, you are only going to have the mother chromosome inside the fetus, and that mother chromosome is going to constitute now of only one grandparent chromosome, let's say the grandmother. So this is called an isodisomy. Is it clear? Now, between the heterodisomy and isodisomy, which one is more severe? Let me ask you the question. The more severe is isodisomy. Is it clear? Because we said that what uniparental disomy is only going to transmit disease when it's from one parent. Is it clear? Uniparental disomy transmits diseases only from one parent. Is it clear? So it means that what? It means that here you see that what? You see that the parent that has transmitted the gene to the child is only the mother. And is it clear? But in heterodisomy, what is nice is that what? The mother had all the gene from the grandfather and the grandmother. It was not from one grandparent. Is it clear? So it's both grandparents. So this patient is going to have a less severe disease, because though it has been transmitted from one parent, who is the mother, both grandparents has completed to produce this particular genotype here. But now in this case, you see that what it is only the mother that has transmitted the gene to her child. Is it clear? To the chromosome to her child, the chromosome pair to her child. And what makes it more severe is that it's not only the mother that has transmitted the gene, but it's only the grandmother again that has transmitted the gene to her child. So this patient is going to have more severe disorder. Now, so that is uniparental disomy. Now the next thing is imprinting. So you need to know that what there are certain. So you need to know that in imprinting, what happens is that what there are certain genes in the body that are imprinted, that are not going to be expressed. Is it clear? There are certain genes that are imprinted. It's another method of genetic inheritance. Certain genes in the body that are not expressed. Now, in cases where they are now expressed, they are now going to cause disorders. Is it clear? So you need the term imprinting is used to describe a phenomenon by which certain genes function differently depending on whether they are maternally or paternally derived. The mechanism of DNA modification involved in imprinting remain to be explained, but it confers a functional change. Is it clear? So this imprinting can work now with uniparental disomy now to produce the disorder. Is it clear? And now imprinting and uniparental disomy, the most common disorder which are present there is Prader-Willi syndrome and Angelman syndrome. Prader-Willi syndrome is a paternal uniparental disomy, while Angelman syndrome is a maternal uniparental disomy. Prader-Willi syndrome means that it is only the father who has transmitted the gene to the child. Is it clear? And it's usually occurring at chromosome 11. So usually occurring at chromosome 15. They have only transferred, the father has only transferred the chromosome 15 to the child. While in maternal uniparental disomy, it is only the mother that has transferred to the chromosome 15 to her child. And you need to know that what is called the term imprinting. Normally, there is a Prader-Willi gene on the father, and when the mother is having the gene that is going to imprint that Prader-Willi gene of the father, is going to close and make sure that Prader-Willi gene is not expressed. If the father is only transmitting now the gene to the child, the mother gene that is going to try to enclose the Prader-Willi gene that was only present with the father is not going to be present again, and then the child is going to develop Prader-Willi syndrome. It's same thing now with the Angelman syndrome. There is Angelman syndrome gene on the mother's chromosome 15. Is it clear? And the father's chromosome 15 also constitutes of a gene that is going to imprint the Angelman syndrome gene, which is present on the mother chromosome 15 chromosome. Is it clear? Now, if there is no father, if the mother has only transmitted a gene to the child, you see that there is no father chromosome 15 gene to imprint the mother Angelman syndrome gene, so the child is going to express Angelman syndrome. Is it clear? So that is with that. The next one is mosaicism. So mosaicism, we have already said that different cells are going to have different cell lines. Is it clear? And it's only from one zygote, and that's the difference with chimerism, where you have different cell lines but from two zygotes. So that's the difference. Fusion of two zygotes. Now, this is a case of a patchy distribution of skin lesion in females with incognitia pigmenti that we have seen later, and we have seen later. We said in an X-linked dominant disorder. We said that it's an X-linked dominant disorder, incognitia pigmenti. So later in males, so usually this X-linked dominant disorder, incognitia pigmenti, is lethal in males but not in females. Why? Because females have a functional X chromosome mosaicism. So that functional X chromosome mosaicism is just what I've just we're just trying to be explaining you every time. We say that certain females, since females have both X chromosomes, well, certain cells are going to express normal X chromosome, while other certain cell types can express the abnormal X chromosome. So they have still a high probability of having a normal phenotype, though they have an abnormal genotype. So like this, in this case with incognitia pigmenti, which is detrimental, which is lethal to the males, females can be alive and still transmit the disorder. Now, this is the trisomy of the chromosome 12b, which exists in mosaicism, as you can see. Now, these are the different disorder pedigree. Now, we have other disorder transmission, other genetic transmission like mitochondrial disorder. Is it clear? We need to know that what not all DNA is contained within the cell nucleus. There are also other certain DNA which are contained within the mitochondria. Is it clear? So the mitochondrial DNA consists of 16,569 base pair that constitute 37 genes. Is it clear? So there are 37 genes in the mitochondrial DNA, and there is some difference in the genetic code between the nucleus and the mitochondrial genome. The mitochondrial genome is almost exclusive coding due to genetic and intervening sequence. No intervening sequence. So actually we have seen that inside the DNA of the nucleus there were what is called the exons and introns, with the terminal exons, introns, and the terminal proteins. Is it clear? And the terminal signals. But here in the mitochondria we have no introns. They only exist as exon in a cyclic pattern, double helix cycle. Is it clear? So that is the mitochondria. So this is the zonal pellucidal with the different mitochondria present. We need to know that what generally mitochondrial disorder are mostly transmitted by the female. Why? Because the female, the oocyte is what contains most of the mitochondria, like 10,000 to 100,000 mitochondria. Why the sperms only contain 10 to 100 mitochondria. So most of the mitochondrial inheritance is going to be due to from mother to the child. So when mother is affected and all the children are affected, it means that it's a mitochondrial disease. Is it clear? When the mother is affected and all the children are affected, it's a mitochondrial disease. But when the mother is affected and all her sons are affected, it means that it is an X-linked disease. Is it clear? So that's the difference. So X-linked disease, you have a difference in the proportion in sex, while mitochondrial and autosomal diseases, all the children are affected in the same proportion. Okay, so these are certain disorder with mitochondrial diseases. So like in the case of Leber hereditary optic neuropathy, so these are the symptoms here, and these are the common mutations that occur in mitochondrial disease, because we say that there are 16,569 base pair that constitute genes. So these are the different common mutations that can be present. We have the MEF, which is the myoclonic epilepsy. Myoclonic epilepsy with red fibers of the skeletal muscle is another one. We have KSA syndrome, so with all the difference, and you have MELAS. So these are the different things that you can visualize in mitochondrial disease. Now, the genetic counseling problem in mitochondrial disease, you need to know that what some disorders of the mitochondrial function are due to nuclear gene mutation. So you have disorder of mitochondrial function may be due to nuclear gene mutation. So you may have even a MELAS that is due to a nuclear gene mutation. You have some disorder caused by mitochondrial mutation are sporadic. So they may not be due to they may vary from time to time. That's the meaning of sporadic. They may not be calculated to really with probability. They're sporadic. That's another problem. When maternally transmitted, not all offspring are affected. Normally, you can see from this pedigree. Normally, in the pedigree of the pedigree of mitochondrial disease shows that when the mother is affected, all the children must be affected. But you see that this mitochondrial disease is sporadic. Sometimes the children are affected, sometimes children are not affected. That's the problem. And then prenatal diagnosis is not feasible with mitochondrial diseases. So all this makes the mitochondrial disease, and also mitochondrial diseases may be encoded by nuclear genes. So it makes the mitochondrial disease to be actually questionable if they are actually even mitochondrial disease or the disease are even coming elsewhere. Is it clear? So that's the problem that we are dealing with mitochondrial disease. So here we are finished with this tutorial, but then we continue with the next tutorial. Thanks for your kind attention. Don't forget to like and subscribe for our channel. Thank you.